Fourscore and 7 years ago, blah, 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 blah. Okay, that works. Okay.
Are these questions what will be on like the big final exam?
Yeah.
Or,
Yeah, I'm on that. That's Kristen.
I'm good. How are you?
Upright, breathing, and movement.
Like 3 hours into the first week, so you can't have that much to be wrong right now. I think I'd like to make it different. The amount of things I feel like
I'm sure you'll see where you can get them.
right now. At this very moment, yeah. I don't know what. Okay, let's go focus my discernment. What do you guys think? Yeah. So many different climates. I do, not very often,
Oh,
but
okay,
you're
you
ready
have
to
that
know.
Your contacts on?
Yes, I do. Yeah. I'm ready. I got it. Yep, that's me. Should I log on that? Thank you for your. Thing would be perfect, but there's.
Join is-
Okay, we're zooming. Emma is supposed to be joining us. Sent her a Zoom link. I hope she's joins us soon. We need to get right with it, so let's go ahead and get started. Lord, thank you so much for warmer weather. It's so nice to not walk outside and be in pain. And thank you for chapel. Thank you for this new week. Wake us up, please. Get us started well on this new week. Thank you for these students, for their dedication to being healers. In Jesus' name, amen. Complement. If you have a handout I gave you about complement, please take it out. So complement was discovered quite a while ago. And when scientists took bacteria and mixed it with serum from animals that they knew had antibodies that bound to those bacteria, the combination of antibodies and serum killed the cells, completely lysed them dead. If you tried to recover those cells on bacteriological media, nothing would grow. The antibodies themselves didn't kill them. They clumped the cells together, but they didn't kill them. The serum by itself didn't kill the cells. There was something about the antibodies plus the serum that killed the cells. So because that something in serum complemented the activity of the antibodies, it got the name complement and the name stuck. That particular activity could be inactivated if you heated the serum. That told the scientists it was probably protein, and they were right. So complement does a bunch of things. Complement, first of all, drills holes in membranes of membrane encased viruses, bacterial cells, metazoan parasites, whatever. So it kills invaders. It's very good at that. Secondly, elements of complement bind to the surfaces of cells or worms or whatever. And they provide phagocytic cells with a little bit of Velcro. So the phagocytic cells can bind to that bacterium and gobble it up. When you put little pieces of Velcro, and I'm trying to use an example, you put a little piece of Velcro on the cell, so now the phagocytes can gobble it up. That is called opsonization. Emma, you're with us.
Hi.
Can you hear me, Emma?
Yes.
How's your knee?
It's okay.
Just okay?
Just okay.
Okay, that's better than terrible.
That's true.
All right. Everybody say hi to Emma.
I am up.
The class says hi, you probably can't hear them. So they hope you recover quickly and come back soon.
I'm hoping the same thing.
Okay, so you'll be on crutches when you come back or just in a boot?
Crutches.
Crutches. Okay,
Yeah.
so we can call you Hop Along. All right. Now, when you put Velcro on cells so that phagocytes can take them up more easily, that's called opsonization. Opsonization, big 50-cent word. Opsonization, you make cells more easily taken up by phagocytes. The third thing complement does is that it augments inflammation by summoning white blood cells. Two of the complement fragments in particular, C5a, C3a, are chemotactic agents for white blood cells. Summon white blood cells. You can do this in culture. You can actually take blood from someone and you can isolate their white blood cells. It's not that hard to do. I could show you how to do it. You know how to do it. It's not hard to do. You can then take isolated complement fragments, C5a, C3a, and you can make a trail of it, and the white cells will go they'll go right towards it quickly too. So these things are quite potent. Now, there are three ways to activate the complement pathway: the classical pathway, the alternative pathway, and the lectin pathway. Classical pathway requires antibodies. Antibodies. The alternative pathway just requires microbes usually, or some sort of invader with a surface that's charged. And then the lectin pathway gloms onto a particular sugar that's rare in our cells, but common in the cell walls of bacteria and fungi, and that is mannose. Mannose. M-A-N-N-O-S-E, mannose. And there's a protein wandering about your blood called the mannose binding lectin. The mannose binding lectin. If it sees a lot of mannose, it gloms onto it, sticks to it, and that will activate the complement pathway. So here's how the whole thing starts. You have this protein called C1. C1 molecularly kind of looks like a snowflake, I guess, a broom, the end of a broom. Students have called it a lot of things, but it kind of looks like these little UFOs over here. And it essentially is a stem with a bunch of ends with these globs on the end. That's C1. C1, if there are antibodies bound to a surface, C1 will bind to those antibodies. So you get C1, the antibody, C1 binds to it, and C1 now becomes a very active serine protease, and it clips some of these other complement components. So it clips complement C2, C4, and then C2 and C4, they're active. They clip C3, and then C3, it becomes an active protease, and then it clips C5, and C5a, C5b. 5a is a very important chemotactic agent. C5b then makes a complex with C6, 7, 8, and 9. And that complex of C5b, 6C, 7, 8, and 9 forms something called the MAC. Now, when I say MAC, I say that to students. Last year, I asked students, what does that mean to you? They said, pretty in one voice, mac and cheese. This is not mac and cheese. MAC stands for membrane attack complex. What C5b, 6, 7, 8, and 9 do is they form a pore. They self-assemble into a membrane and they form a pore. You can actually take detergents, put it in a test tube, throw these proteins in, and they'll make little pores in the detergent. And you put a pore in a membrane, what happens to the cell? Come on, guys, I put a pore right here in your tummy. What happens? Your insides leak out, right? Is that good? No, bad, very bad. You'd probably die, right? Yeah, if you weren't treated, you die. So cells get lots of holes in them. What happens to the cell? It gets the deep six. It dies. And if it's an invading bacteria, what do you say? Good riddance. That's a classical pathway. You have to have antibodies for the classical pathway. It depends on C1. C1. So C1 binds antibodies. Basically, you activate a bunch of components and you culminate in the formation of the MAC. You kill the cells. That's really all I want you to know. The dirty details of this, we'll go over that next year in micro.
What does MAC mean?
Membrane attack complex, not mac and cheese. Membrane attack complex. That's why it's called MAC. The lectin pathway starts with the lectin, mannose binding lectin. And guess what? Molecularly, the mannose binding lectin looks a lot like C1. It looks like a little cleaning brush, molecular cleaning brush. But instead of binding antibodies, it binds mannose, large stretches of mannose. Where do you find large stretches of mannose? In the cell walls of bacteria and fungi. And then the same thing happens. You generate the MAC and you kill the fungus or the bacterium or the whatever. So the lectin pathway, classical pathway, pretty similar. Classical pathway, you need antibodies. Lectin pathway, you need, say it, mannose. The alternative pathway is a little weird, and it goes like this. C3 is just always being activated a little bit inside your bloodstream. Normally it gets activated, doesn't have anything to bind to, and just goes away. But if cleaved C1 has a bacterium, it'll stick to the surface of the bacterium. And if it sticks to the surface of the bacterium, it recruits a bunch of other proteins to it, and they activate the MAC, and that kills the cell. So the alternative pathway is C3 activated, but only if C3 is able to alight on the surface of an invading cell. So one more time. Classical pathway, you need, say it, antibodies. Lectin pathway, you need mannose. Alternative pathway, you need an invading cell. All roads lead to Rome. Rome in this case is the MAC. Okay, understand? Now, can complement attack your cells? What do you think? You have an autoimmune disease that makes antibodies to your own cells, can complement damage your cells? Yeah, it can. It can. And it can do a lot of damage to your cells. So that's something to keep in mind when we talk about autoimmune diseases. Now, one more thing before we move on to some other stuff. So we talked about the clotting cascade. Here's the clotting cascade. Turns out that our buddy, factor 12, that starts, which one? Intrinsic or extrinsic clotting pathway? I don't speak mumble. Intrinsic. Good, Kristen. Good, Kristen. Pat yourself on the back. Intrinsic. It turns out that 12A can also activate this thing called the kinin pathway. The kinin pathway. What's the kinin pathway? The kinin pathway starts with an inactivated enzyme in your blood. By the way, there's a term you may have heard. If you haven't, you'll hear it now. If an enzyme is made in an inactive form, and then it later gets activated, that inactive form is called a zymogen. So zymogen, it's an enzyme that's made in an inactive form. It needs to be activated, usually needs to be clipped. The front of it or the back of it, or needs to be clipped in the middle or something like that, then it's active. Zymogen. So prekallikrein is a zymogen, and it is converted to the active form by our old buddy, activated factor 12. Kallikrein is now an active protease, and one of its targets is this molecule here. This is high molecular weight kinin. Big protein. Kallikrein clips it to a small little peptide called bradykinin. Brady in Greek means short. So you take kinin, you make bradykinin, short kinin. Bradykinin does two things that are really important. Number one, it's a potent vasodilator. Vaso, period. Oops, what did I just do? Vasodilator. Where'd my V go? Oh, good gravy. Huh. Because if I write vasodilator, I'm going to make a mess out of it. The other thing bradykinin does, and kinins as a family, there's a family of little peptides called kinins. Kinins activate nociceptors. What do nociceptors detect? There you go. Reagan's absolutely right. Pain. They activate pain. So they cause vasodilation and they activate pain. So good gravy. Vasodilation. It keeps disappearing. Now, kallikrein is also going to take this zymogen, plasminogen, and clip it to plasmin. Plasmin, you will notice, does two things. Plasmin can actually activate the complement cascade because it will clip C3 into C3 and C3A. Plasmin, however, usually is used to degrade fibrin. Now, typically, plasminogen is converted to plasmin by this protein called tissue plasminogen or tissue plasmin activator, or TPA. And commercial versions of TPA are used to degrade clots that you get in your brain or in your heart or other places. If you have dialysis tubing, you get a clot in it, you can use a version of it to actually clean out the dialysis tubing as well. So that's available as a commercial product, but plasminogen can be converted to plasmin also by kallikrein. So this again is going to help with the inflammation cascade as well. Thrombin also, and I didn't mention this last time, turns out that there are receptors on cells for thrombin. Those receptors are called PARs. There are thrombin receptors on platelets. There are thrombin receptors on other cells. When thrombin binds those receptors, it will augment inflammation. There are drugs that actually block those receptors. Vorapaxar is a drug that's used if you get a stent. It blocks those PAR receptors. So the platelets can't be activated by thrombin. And when cells are damaged, there's another set of molecules that are made that also augment inflammation, and those are these molecules called prostaglandins. So prostaglandins are made from a fatty acid found in your phospholipid membranes. That fatty acid is a C20 fatty acid. It's called arachidonic acid. Arachidonic acid. You've got lots of arachidonic acid in your cell membranes. So arachidonic acid's not an issue. When cells get damaged, you activate phospholipase A1, phospholipase A1 takes your cell membrane phospholipids, it clips that ester bond between the glycerol backbone and the arachidonic acid. And then arachidonic acid is a substrate for this enzyme, cyclooxygenase. And cyclooxygenase converts arachidonic acid into these prostaglandins. And these prostaglandins are pretty universally pro-inflammatory, except for this one here, prostacyclin. Prostacyclin is anti-inflammatory, but the rest of them are pro-inflammatory. Now, if you notice here, phospholipase A1 is inhibited by steroids. That's the reason why steroids inhibit inflammation. They prevent the liberation of arachidonic acid from membrane phospholipids. And over here, cyclooxygenase is inhibited by this group of drugs called non-steroidal anti-inflammatory drugs, of which aspirin is a member, of which ibuprofen is a member, of which naproxen sodium is a member, of which Solindac is a member, meloxicam is a member, piroxicam is a member, indomethacin is a member, blah, 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 all the different NSAIDs. That's how they quell inflammation. They prevent cyclooxygenase activity. When you prevent cyclooxygenase activity, you prevent prostaglandin synthesis. Oh, and looky here, leukotrienes. What are leukotrienes made from? They're made from arachidonic acid. So these leukotrienes are made by lipids. They're also made by neutrophils. And we talked about what they do. You can see this very nicely here. They're chemotactic. They cause bronchospasm, so they're important in asthma. They cause vasoconstriction to prevent blood loss, but they cause increased permeability. They cause the cells of the endothelium to not stick together quite so well, so the vessels get leakier. And then damaged cells, endothelial cells, epithelial cells, and also white cells, neutrophils, eosinophils, so on and so forth, macrophages, make these small proteins called chemokines. Chemokines are signaling molecules. And they essentially say, white blood cells, come, 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 come. Do you guys Saturday hear the siren go off? So the fire department, they have the siren, and the siren is meant to go off if there's a tornado or something like that. Every month, they're supposed to run the siren so they know it works. The siren runs the first Saturday of every month. You can set your watch by it, 12 o'clock, and it goes for a minute. And if you're really close to the fire department, it goes off, your ears start ringing. And that's essentially what a chemokine is like. It's a siren. And the white cells come running towards the siren. Cells have receptors for the chemokines. When the chemokine binds, it means I move in the direction of the chemokine. So it's kind of like the story of Hansel and Gretel and the breadcrumbs. That's how we get home. We follow the breadcrumbs. Okay, I know the birds ate it and they ate the breadcrumbs in the story, but you still get the point. And this picture just shows you essentially the cells move in a gradient direction. So gradient means there are more chemokines as we go this way. Cells are going to move from less to more. They move from less to more. And some cells that they experience chemokines, they will actually express their own signaling molecules, the most prominent of which are cytokines. And let's talk now about cytokines. So there are 5 main types of cytokines. There are the interleukins. Interleukins essentially act as communicating molecules between white blood cells and non-white blood cells. They promote the development and differentiation of lymphocytes, B and T lymphocytes. One particular interleukin I think is pretty important to mention. That is interleukin 2. Interleukin 2 is going to be very prominent when we talk about adaptive immunity. This is a T cell growth factor. T cell growth. Come on, draw you. Growth. I'll just say GF, growth factor. Another group of cytokines are the interferons. Interferons tend to be made, first of all, by virally infected cells. They signal to nearby cells that viruses are about. And so now these nearby cells modify their cellular machinery to make it much harder for the viruses to infect them. That is the case for the so-called type 1 interferons. Made by virally infected cells, signal to nearby cells to modify their molecular machinery so that viruses have a much rougher time dividing in them. The type 2 interferons, I'll just say T2 because, boy, man, doing this with a mouse is hard. Type 2s. The type 2s are made by T helper cells, and they essentially supercharge macrophages so that now macrophages can kill just about anything they get their pseudopods on. So type 2 interferons are immune system, immune response augmenters. So interferons, interleukins. We also have the other one we talked about, tumor necrosis factor. We've already talked a bit about tumor necrosis factor, so I won't say a whole lot about it here. And the one that's important for inflammation is tumor necrosis factor alpha. Tumor necrosis factors in general, they do two things, well, two big things. They tend to signal to damaged cells to undergo programmed cell death. They also will initiate inflammation, so they cause fever. They increase expression of adhesion molecules, so immune cells can attach and move toward places, and they increase vascular permeability. And they're made by lots of different damaged cells. So interleukins, interferons, tumor necrosis factors. We have two others that are types of growth factors. Those are the colony stimulating factors, the CSFs. That stands for colony stimulating factors. These go to the bone marrow. They bind to proliferating cells in the bone marrow. They stimulate precursor cell proliferation and differentiation. They stimulate differentiating cell proliferation and differentiation. So there are colony stimulating factors for macrophages, for neutrophils, for red blood cells, for eosinophils, for what have you. Those are the colony stimulating factors, the CSFs. There are commercially available CSFs that are used as drugs too, that you will probably give to people during your careers. And finally, there are the transforming growth factor group of cytokines. Now, this particular group of growth factors are anti-inflammatory. These downregulate inflammation and they promote healing. So as inflammation goes on, TGFs get made at higher and higher and higher rates, causing inflammation to go become less and less and less prominent. And they are important in transitioning inflammation to healing. All right, pause. Questions? Guys are writing furiously. That's good. That's good. So cytokines bind to receptors. They bind to receptors. Those receptors initiate a signal transduction pathway inside the target cell that leads to changes in gene expression. The different cytokines produce different effects. And some of these are very pro-inflammatory that you can see here. And some tend to regulate the immune response. And boy, howdy, looky here. Look at this one here, TGF beta. So these here, they regulate the immune response. Interleukin 4, interleukin 5, interleukin 10. Interleukin 10 is a big regulator of the immune response. And then these over here, which tend to be more pro-inflammatory. And this slide I got from a manufacturer. They make proteins, interleukin proteins that are purified that you can buy for an exorbitant price and use in your experiments. I thought this slide was really cool, but it just kind of shows you what some of these do. And the thing I want you to see about this slide, you don't have to remember stuff on this slide except for really specific stuff I might mention. Notice how a lot of them work in combination with each other. So activating one particular receptor gives you one response, but if you activate two of these receptors, it gives you a very different response. And that is something that you're probably saying, you're studying the endocrine system by now, right? Or you have studied it. Okay, been there, done that, got the hat. You probably notice that hormones can sometimes work together. If they work together, they can either augment each other, they can fight each other, or they can work synergistically sometimes. They almost work in a you scratch my back, I'll scratch yours kind of way sometimes. So these dual signals can cause cells to have very different responses. Oh, one I want you to be aware of, this particular cytokine right here, interleukin 8. This is a chemotactic factor for neutrophils. That one's pretty important. So that's one I want you to know. And then the interleukin 1 and 6 and tumor necrosis factor, they're the early ones that initiate the acute phase proteins from the liver. I want you to know those two. The rest of them, some of these, we'll learn more about these next year in micro, when we go over the immune system in micro. All right, a word about nitric oxide. Nitric oxide is typically made by the endothelium, and nitric oxide it's essentially a signal that keeps the smooth muscle here relaxed, and it essentially tells the bloodstream everything's Irie. We're fine. I'm okay. You're okay. Let's relax. That's what nitric oxide does. Nitric oxide is made from the amino acid arginine. Those of you who are athletes, have any of you ever, there are these pre-workouts you can get that are full of arginine? Have any of you ever tried them? Will any of you admit to having tried them? All right, let's try it this way. Do any of you have friends who've tried them? Did they get a headache? Yeah, because lots of arginine causes lots of nitric oxide, causes vasodilation on the vessels in your brain. It can cause a headache. And plus, some of these pre-workout concoctions have a lot of arginine in them. Oh my goodness. Some of the stuff they sell at GNC. Yeah, that'll give you a headache, all right. Okay, story. I had a student, I was in the gym, and the student of mine, he comes up to me and he says, can you check out my buddy here? And I walked over, he was beet red. And I said, I bet I know what's wrong with him. I said, you take a pre-workout drink? He said, well, yeah, just this. I said, let me say it. It's full of niacin. Niacin will cause you to flush. About 20 minutes after you take it, it'll cause a flush. And some of these pre-workouts have a lot of niacin in it. So you'll be beet red. And I was so tempted. I wanted to say, oh, gee, this is bad. We need to call an ambulance. I was so tempted, but I didn't. Anyway, I said, it's the niacin in here. It'll go away. You're fine. He said, it really itches. I said, I know. If you were going to take this stuff, take half an aspirin, a half an hour before you take it, then drink it. You'll be fine. He said, oh, thanks, man. Oh, I was so tempted, but I didn't. So that's nitric oxide. Now, nitric oxide is also used by macrophages, and it combines the nitric oxide with the hydroxyl atoms that it makes to make peroxynitrites that are really, really nasty. And these will kill microbes very effectively. And that's why arginine is in these pre-workouts. The theory is that it increases blood flow to the skeletal muscles and helps you work out better. Between you and me and that door, pre-workouts are a waste of money. We actually tested a couple of pre-workouts here. A bunch of HES students said, let's test this stuff. So we tested endurance, we tested power, we tested reps, that and placebo, because it comes as a fruit punch. So we just made Hawaiian seafood punch. Hawaiian seafood punch worked as well as these pre-workouts, and it's a lot cheaper. Save your money. Aaron?
I think most of the part of it is the focus.
But the caffeine in it to jack you
I
up?
don't know what it exactly is, but like people get like super focused and can't. I don't know what.
Then drink coffee.
I've heard.
It's cheaper. Seriously, go to Big B or the new place that opened down there. What's it called? Yeah, I hear their coffee is really good. I don't drink coffee, so I hate it. I hate coffee. Oh, I just yeah, my kids all like it. My wife likes it, but she can't drink it anymore. Just drink coffee. I'll do the same thing, and it's cheaper. Anyway, platelet activating factor is a lipid, and it's made by platelets, and it binds to a member of the G protein coupled receptors, and it causes mobilization of calcium stores inside cells, and it does all these cool things to platelets, and it can activate other cells too. So that's another molecule made by platelets that helps augment inflammation as well. Now, we talked about these pathogen-associated molecular patterns that are on the surfaces of invading pathogens. You have receptors on the surfaces of white cells that bind to these pathogen-associated molecular patterns. They are called pattern recognition receptors. So when the kitty cat invades, it says, her. The kitty cat invades, has all that fur, and the fur binds to the her. Okay, it's stupid, I know, but it helps you remember it. Then you'll thank me. So there are two groups of pattern recognition receptors. One group are signaling, for signaling purposes. The other are for phagocytosis purposes. So the class of pattern recognition receptors, PERS, that are for signaling are called toll-like receptors, TLRs, toll-like receptors. You have two types. You have toll-like receptors on the surfaces of cells. They detect extracellular pattern-associated molecular patterns. But remember, some pathogens are sneaky. They get inside your cell. So you have intracellular toll-like receptors for binding pathogen-associated molecular patterns. These are internal toll-like receptors. And the toll-like receptors were named by biochemists. So they're TRL1, TRL2, TRL3, TRL4, 5, 6, 7, 8, 9, so on and so forth. So the ones that are internal are 3 and 7 and 9. The rest of them are extracellular. And when you activate the toll-like receptors, here's what happens. You make cytokines. You make interleukins, interferons, chemokines, and what have you. And that activates the immune response. So let's do a brief survey of some of the immune cells. We talked about a few of these before. Mast cells, remember, are found in loose connective tissue. They are very much like basophils, but it's clear from lineage studies they're not related to basophils lineage-wise, but they functionally are a lot like basophils. So these granules are full of histamine, leukotrienes. When they are activated, they degranulate. And they release all that good stuff, causing the events we talked about before. So they cause vasorelaxation, they cause the endothelial cells to contract, they cause the vessel to become leaky, and so on and so forth. So platelets normally are round when they're activated. They get these spiky dude extensions of the cell. Look at how these platelets are all bound together. They make those receptors that allow them to aggregate because they make the glycoprotein 2B3A receptors that bind to fibrin. And now the phagocytes. These are phagocytic cells. They gobble things up. So the neutrophils, these are first on the scene. The granules are loaded with things that will kill microbes when they gobble them up. These cells, as we said before, they don't live very long because they're so actively phagocytic, they eventually kill themselves. So they give their lives for your immunity. There's a sermon illustration in there somewhere, but I'll save that for later. Eosinophils. Now, how did these things get their oddball names? They got their oddball names because of a stain that you use in tissue called hematoxylin and eosin stain, affectionately known as H&E stain. Very easy stain to do. I can take you to the lab, show you how to do it. You know how to do it. It's not hard. Eosinophils stain, their granules stain with eosin. Because they stain with eosin, their granules are pink. Eosinophils play a role in allergies and in parasite infections. When metazoan parasites are in your body, eosinophils surround it and let loose their granules, and the worm will, that's not good. And eventually, if they keep at it, the worm will just dissolve. So important for allergies and parasite infections. Basophils. Functionally, these are a lot like a mobile mast cell. So they have granules full of histamine, full of heparin, full of leukotrienes, and they're called basophils because they stain with hematoxylin, their granules, and their granules are deep ocean blue, almost purple. And then the monocytes. Monocytes don't have granules, but they have these big mahunka nuclei. And these cells are big, 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 big. So you can see the cytoplasm really doesn't have any granules, unlike these other cells that are very obvious granules when you stain them. And when a macrophage leaves the bloodstream, gets into the tissue, it becomes a macrophage. And a macrophage is an activated monocyte. It has granules, but you can't detect those with stains. So it is an agranulocyte. Some macrophages will mature and become dendritic cells. Dendritic cells. Dendritic cells, as you see, have lots and lots of tentacle-looking extensions from the cell. These cells are exquisitely important at identifying foreign substances in your blood, in your extracellular fluid, in your lymph, grabbing that foreign substance, processing it, and then presenting it on the surface of its membranes. When it presents it on the surface of the membrane, essentially, the immune system has been given the go-ahead to respond to this foreign substance. So the dendritic cells are really important for that process. And they are derived from macrophages. They're highly motile, very phagocytic, but eventually they settle down, they get those processes, and they just sample things that come by and present them on their surfaces. So dendritic cells are important in what we call antigen presentation. Very important, antigen presentation. So the process of phagocytosis, you have heard this before probably in the first semester A&P, so I'll go through this very quickly. You have usually a receptor ligand recognition event. Then the membrane starts to ruffle, it moves around the entity, you get extensions of the cytoplasm that are cytoskeletal mediated. They move around the object, the membrane fuses, and you form a vesicle called a phagosome. The phagosome then fuses with a Golgi apparatus-derived vesicle called a lysosome. That's called phagosome-lysosome fusion. The phagolysosome, as it's now called, digests the material that was in the phagosome. If it's things the cell can use, the cell takes it up, uses the goodies as it needs. Then you have a residual body. Things that can't be digested are expelled by exocytosis. Phagocytosis is augmented if the cell, if the entity has things on it that receptors on the phagosome can bind. And so antibodies or complement particles provide things that the phagosome membrane can bind. So the antibodies or the complement fragments are called opsonins, and that process is called opsonization. Greatly facilitates the process of phagocytosis. And so this shows you all the things that initiate acute inflammation, tissue damage, invasion of the damaged tissue by bacteria, and what have you. We talked a lot about that already. We talked about what happens with the dilation of the blood cells. Let's talk a bit now about healing. Fibrin, it turns out, is an excellent surface for fibroblasts to attach and migrate. So once you make this fibrin plug that prevents bleeding, you now have cells that are going to move down this fibrin In fact, in laboratories, certain cells you can buy. You don't buy fibrin, you buy fibrinogen and you use it to make fibrin. You can put fibrin on your culture dishes, and certain cells migrate incredibly well on fibrin. It's very good for that. Now, the fibrin plug gets degraded by activated plasmin. But these cells are two groups of cells that'll migrate this way and start to repair the tissue. First of all, there are fibroblasts. You've got fibroblasts all over the place. Many of them are just sitting in your tissue, asleep. They're taking a nap. Can you guess what wakes them up? No guesses what wakes them up? One of the growth factors released by platelets is platelet-derived growth factor. Another growth factor released by platelets is fibroblast growth factor. Both of those serve to bind to receptors on the surface of the fibroblast and wake it up. So the fibroblast wakes up, divides, migrates to the area of damage, starts to lay down collagen to repair the tissue. Now, the other group of cells that move in are EPCs. What are EPCs? EPCs, that sounds like something our plumber would use to repair our pipes. Well, kind of, sort of. Endothelial progenitor cells. Endothelial progenitor cells come from bone marrow, but they also come, there are some of them hanging out near blood vessels. They will divide and migrate, and they will start to make new blood vessels to repair the ones that were damaged. And then the epidermis, keratinocytes will move along this fibrin. They will close the wound like that. You get this new granulation tissue. That new granulation tissue is full of new blood vessels that are being made to provide oxygen to the area. That's due to the fact that the platelet released VEGF, which is vascular endothelial growth factor. The granulation tissue is then replaced by new blood vessels and new tissue. And that essentially is how you get healing. So look at the things that are important in healing. Platelet-derived growth factor, TGF-beta, that's released by lots of damaged cells, lots of white blood cells, VEGF, that's released by platelets. And then the other group of cells, very important in healing, are macrophages. When macrophages move into an area, remember, they're the latecomers. Neutrophils come first, and macrophages. They're very, very phagocytic, very pro-inflammatory. They have what's called an M1 phenotype. That M1 phenotype, after a while, gives way to an M2 phenotype. And the M2 macrophages are pro-healing. Anti-inflammation, pro-healing. And so those cells, as you see, will promote proper healing. Now, healing can sometimes go wrong, where you don't get enough collagen. You haven't cleared the area out well enough. Or maybe you get too much deposition of collagen or something like that. So healing sometimes can go awry, particularly in people who have mutations in collagen deposition. So people with something like Ehlers-Danlos syndrome, or people with Alport syndrome, or other types of collagen deposition issues. And this abnormal wound healing can cause either scars because scars, regular scars will tend to contract and be very small. You can get hypertrophic scars, too many scars, or you get a so-called keloid. And keloids essentially are these large things that don't really contract. And that's due to abnormal wound healing. Keloid scars are seen most often in wounds that get re-injured repeatedly. And that's why folks who self-injure, they self-injure in the same place, they can get keloids. And keloids, they can crowd out normal tissue, prevent the normal tissue from doing its job. All right, let's go back to there. And yeah, they can see. All right, so I want to, no, discard you. All right. Okay, yes, I know it's been, yes, thank you for telling me. All right, in the 3 minutes we have left, we'll just get to start this. Let's talk about adaptive immunity. So adaptive immunity is the other arm of the immune response. Adaptive immunity is slow. It's very specific. And it's like an elephant. It has a long memory. So innate adaptive immunity, think tortoise, hare. Innate, hare. Adaptive immunity, tortoise. Adaptive, slow and steady wins the race. The hare, oh, I'll just burn it up as fast as I can. The big elements of adaptive immunity are the lymphocytes. B lymphocytes, T lymphocytes. The B lymphocytes, as we will see, will secrete these glycoproteins called antibodies. Those antibodies bind specifically to foreign substances in the body against which you mount an immune response. Those foreign substances are called antigens. Antigens. Foreign substances against which you mount an immune response are called antigens. And because the adaptive immune response generates cells programmed to fight a specific antigen, those cells, after a robust immune response, become quiescent and they become memory cells. That's the reason why vaccination works, because the adaptive immune response has memory. That immunity can take two forms, passive and active. When you guys do your clinicals, you have to have all your vaccinations. So you either have an attenuated live virus injected into your body, and your body mounts an immune response against this poor growing virus. That's an active immunity response. Or in the case of your COVID-19 shot, you got the synthetic RNA that was encapsulating these nanoparticles and then engineered your cells to make these viral antigens. You responded to it. That's active immunity. Mother breastfeeds her baby. The baby gets secretory antibody through the breast milk. Did the baby make the antibodies? No. Does the baby have the antibody? Yes, they were given to the baby. That's passive immunity. Passive immunity. The baby receives IgG antibodies through the placenta. Those will serve the baby for about the first 6 months of life. Did the baby make those? No. The baby got them from the mother. That's passive immunity. You get a shot with antibodies to deal with, say, snake venom. You're not immune to the snake venom. Well, good for you. But that's passive immunity. You didn't make it. It was given to you. Now we're out of time. Very good. So We will pick this up on Wednesday. Oh, by the way, the case studies that we do in lecture, because I don't have time to go over them, there is a file here. Lecture case studies. If you go to those, the answers to those case studies are there. So you can click on those and you'll get the case study will come up and it'll actually give you the answers. So it gives you the correct answer and then it tells you essentially how the cow eats the cabbage on that and blah, 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 and so on and so forth. And yeah. All right, Emma, heal thyself. We're going to knock off, okay? So you have a good day, all right?
All right. Thank you for letting me zoom in.
Oh, absolutely. So we'll Zoom Wednesday too, okay?
Yes. Thank
All
you.
right.
Have a good
Okay,
day.
Lord bless, dear lady.
Bye.
Tell Emma bye. Bye, Emma.